Implantation of the blastocyst usually occurs 6 to 8 days after fertilization. By the end of day 8, the blastocyst has burrowed into the endometrium of the uterus. At this time, it is composed of two main components: the outer cell mass, the trophoblast, and the inner cell mass, the embryoblast. As the trophoblast makes contact with the endometrium, it differentiates into two layers: an inner cytotrophoblast and an outer syncytiotrophoblast. The embryoblast differentiates into a bilaminar embryonic disc composed of two cell layers, the hypoblast and the epiblast. Soon after the embryonic disc has formed, a cavity begins to appear between the epiblast and the cytotrophoblast, known as the amniotic cavity. Cells originating from the hypoblast begin to migrate, forming a thin membrane, which covers the inner surface of the cytotrophoblast. This is called the exocelomic membrane. The exocelomic membrane and cells of the hypoblast together form the walls of the primitive yolk sac. By day 9, the blastocyst is completely embedded in the uterus wall. At this stage of development, the growth of the syncytiotrophoblast and cytotrophoblast is much quicker than the bilaminar embryonic disc. Small holes called lacunae begin to form in the syncytiotrophoblast as it continues to expand. By day 12, the lacunae stop growing and fuse to form large interconnecting spaces called lacunar networks. Capillaries in the endometrium surrounding the developing embryo dilate, forming maternal sinusoids. As the syncytiotrophoblast continues to expand, enzymes begin to erode the lining of the sinusoids and uterine glands, allowing maternal blood and uterine secretions to flow into the lacunar networks, establishing a uteroplacental circulation. The blood and uterine secretions only come into close proximity to the embryo, allowing the exchange of gases and metabolites. Around the same time, a new population of cells appear between the inner surface of the cytotrophoblast and the outer surface of the primitive yolk sac, known as the extraembryonic mesoderm. Large cavities begin to appear in the extraembryonic mesoderm. These gradually fuse to form one single cavity, called the chorionic cavity. Around 13 days after fertilization, a large portion of the exocelomic cavity is pinched off, forming a smaller cavity, the secondary yolk sac. By the end of the second week of development, the chorionic cavity enlarges, and the bilaminar embryonic disc is joined to the trophoblast by a band of extraembryonic mesoderm called the connecting stalk, the future umbilical cord.
During the third week after fertilization, the three primary germ layers are formed, which provide cells for organ formation in the following months. These germ cell layers are formed by a process known as gastrulation, which involves rearranging epiblast cells. As cells from the epiblast migrate, a faint groove, called the primitive streak, forms on the surface of the embryonic disc. With the formation of the primitive streak, it is possible to recognize the head and tail ends of the embryo. Some epiblast cells migrate, differentiate, and then replace the hypoblast to form the endoderm germ layer. Other epiblast cells migrate and differentiate to form a new layer between the epiblast and hypoblast, identified as the mesoderm germ layer. The remaining epiblast cells become the ectoderm germ layer. The result of gastrulation is the formation of the trilaminar embryonic disc, consisting of the three germ layers. The ectoderm gives rise to all nervous tissue and the epithelium of the skin. The mesoderm gives rise to blood, muscle, bone, and connective tissue. The endoderm forms into the epithelium of the gastrointestinal tract, the respiratory system, and many organs. Further differentiation of mesodermal cells begins the formation of the notochord. The notochord is important in starting or inducing the specialization of other cells, including those directly above them, called the neural plate. Epiblastic cells in the neural plate are induced to begin to mound up and fold into a groove. The neural groove continues to fold. Adjacent notochord mesodermal cells are induced to form somites. Somites are the eventual source of cells that form muscle tissue, skeletal, and other connective tissue. The neural groove completely folds inward to form the hollow neural tube. The neural tube arises and its cells eventually develop to form the brain, spinal cord, and other parts of the nervous system. Also during the third week, mesodermal cells from the primitive streak rearrange to form blood vessels in the embryo, and a primitive heart tube is formed. Continued development of the chorion results in the formation of the chorionic villi, projecting into the endometrium to form an exchange area called the intervillous space. The maternal blood vessels and fetal blood vessels remain separate, so maternal and fetal blood do not normally mix. However, oxygen, antibodies, nutrients, and waste products are all exchanged via the intervillous spaces.